As many of you may know, I am disabled. I have an aide who helps me sometimes. And today, as we were going out to my car, this is what I found. When we saw it at first, I wondered, what in the world is this? It's not any time in the year that it could be pollen. It's way too late in the year for that. And as I looked at it, I realized there were long strands that it was connected to. And I noticed that where the little spots were not, there had been many, many long strands. I recognized this pattern and I immediately knew what it was. I said to my aide, "That looks like desiccated chemtrail." She looked at it and she went to touch it and scratch it off of the car a little bit. I said, "Don't touch it. That stuff is deadly. Don't touch it. That could have nanites in it." So I went back in the house and I looked up desiccated chemtrail. And this is what came up. Chemtrail rain microscope analysis. Look at the long fibers that the tiny particulates are attached to, some of them. And the next thing I came upon was a dry particle storm. This is a chemtrail storm at night without any wind in Germany in 2008. I saw numerous storms like that while we were on a trip this weekend. It looked like rain. It was during the day, but it was white. And the person I was with said, "You know, that's rain." And I said, "Sweetie, that is white. Rain is gray when it comes down." You'll notice in this dry particle storm that things are flying around it and very much alive, very magnetic. There's a lot of magnetodynamics involved. And as you see, this is not bugs and things that are flying around. It's all chem fluff. Then I saw this. Doesn't that look familiar? That looks exactly like was on my car. You see? Tubular fibers. And here it is again, tubular fibers. This was taken out of a person's body. This was taken out of a person's body. The same thing that was on my car. And I said, you know, the color that it is looks like desiccated blood. I had heard that there was desiccated human blood in some chemtrail, and I became infuriated. There are also hexagons and crystals found in chemtrail, as well as taken from people's body that have morgellons in their body, in their blood and in their flesh, particularly out of lesions that come from this illness, if you want to call it that, this syndrome that results from chemtrail. Nano-array devices are found in these people that are found also in chemtrail. And I became infuriated because I knew, as you see here, it's very obvious, these are filaments that contain red blood cells. Samples have been taken from the environment, from people. A lot of research has been done, and I want to share some of it with you.
The second group of materials found in these environmental samples is unidentifiable fibers. And I really want you to appreciate the meaning of unidentifiable. These fibers have been sent to sophisticated laboratories, and there is nothing in the databases that match them. So these are fibers, we could say, that do not exist in nature. People around the world are developing lesions on their skin that ooze and produce fibers. This is known as Morgellons syndrome. Tissue samples cultured from ordinary people without this ailment contain and grow the very same fibers. Here's a 5-inch lesion on a woman's body that has never healed, 5 inches. The fibers or filaments are actually tubules with hollow insides. When these fibers are cultured, they produce colonies of filaments. You can see the extension filaments. And these colonies continue to grow and reproduce, branching out into more filaments and more colonies. The filament cultures can be grown from saliva samples, tissue from the skin, mucus, urine, blood, from animals and people, regardless of the presence of the Morgellons condition. The fibers are segmented with visible structures inside them. So where do these fibers or filaments come from? Airborne environmental samples that were collected by Clifford Carnicom, he is a researcher in this subject, in 2000, the year 2000, gathered at high altitude on a mountain in New Mexico, showed the presence of those fibers whose structure matches exactly the tubular filaments. When I say fibers and filaments, basically interchangeable, showing up in our blood, tissues, and skin. Additionally, the samples collected by Mr. Carnicom showed what he calls, and was called in biology, desiccated erythrocytes. This is a multisyllabic term, but it means dried red blood cells. So, why these were in the air? Why are dried red blood cells in the air? Very puzzling. A medical microscopist, a biologist specializing in microscopy, confirmed to him that they were human red blood cells, but that they had been engineered in some way so as to be preserved. So again, you have to ask yourself, if I were 5 years old, I would say, Mommy, what are red blood cells doing falling out of the sky?
Look at these things. They're exactly what was on my car. Look at this. And there were millions of them, front, back, sides, everywhere. Desiccated blood on my car. The first thing I thought was, okay, you people want us dead. But I think there's more to this agenda.
So let's go back to the filaments and the structures they contain, these ones that Clifford Carnicom has found. In those unidentifiable filaments, he has observed the formation of red blood cells and submicron-sized structures. So now you've got a filament making its own red blood cells. The engineered cells, the red blood, very, very tough, withstanding excesses of heat and chemicals, indicating that they are designed to endure almost anything. He has put them in a Bunsen flame, he has poured bleach on them, acid, and they still endure. In addition, they are able to replicate, growing outside of the body in a petri dish. This is highly sophisticated technology going on by itself, not in a laboratory. Here you have a nanoarray that she found, a couple of them in her body. Nanoarray is a tiny device used in biotech for DNA hybridization. And on the left, you see a diagram from an industry website of a nanoarray. And on the slide, you can see actually two of them. She's marked them with red X's that came out of her body, looking exactly like this. And again, I'll say it again, the nanoarrays are these tiny devices that are used for DNA hybridization. Now here are the nanotechnology dyes. And there you see the colored plaques that have been coming out of people's bodies. One on the below has fibers attached to it. The industry tells us that quantum dots are tiny nanocrystals whose small size gives them unprecedented tunability. Okay? Tunability. The piezoelectric effect is the internal generation of a voltage when an outside pressure is applied, and vice versa. You can Google the piezo effect. The piezo effect occurs in crystals, ceramics, DNA, and certain proteins. So an example is, when you put a frequency to a crystal, it generates a voltage. And when you put a voltage to a crystal, it responds with a frequency. We call this piezoelectricity, electricity resulting from applied compression or frequency. Recent work by Clifford Carnicom reveals that a filament culture subjected to a blue light frequency, that's 375 nanometers, resulted in explosive growth of the culture within 24 hours after the initial incubation. So he put the blue light on, incubated it for 5 to 7 days, and then it exploded into all those little fibers that you see. We now live in a frequency-filled world. I don't have time to address HAARP, artificial weather, and the enormous spread of unnatural frequencies that are affecting the Earth today. What is being done to our biology? Again, I'll repeat Ross 80's little tenet here. Chemical bonds are magnetic bonds. Electromagnetism is capable of changing what is happening in our bodies. Frequencies are capable of supplying to the synthetic materials in our bodies the force or power that activates them, gets them working, makes them come alive. Nothing about us today is normal. We have hair that glows and skin that shimmers. This is from a healthy woman in Oregon who's been doing microscopy. And here's a hexagon that grew a fiber in 20 seconds as it was being observed under the microscope. On February 21st of this year, Time magazine had a feature story on the singularity, the term signifying the merging of man with machine. The story was released on Valentine's Day, February 14th, to underscore a love affair, the marriage of humanity and technology. The article tells us that a transformation is coming, and our species, Homo sapien, will no longer be recognizable as itself. We will be something new, something better. The time predicted for this transformation is 2045. The man who is making this prediction is Ray Kurzweil, a futurist known for his uncanny accuracy in just this area, the pace at which technology grows and improves, such that it will one day be smarter and better than us. That's the singularity. There's a Singularity University hosted by NASA, sponsored by Google, to teach people about the intelligence explosion. Ray Kurzweil has made fortunes over and over as an engineer and inventor. A documentary about him is called The Transcendent Man. He wrote the best-selling book that you see here, The Singularity is Near, which came out in 2005. Singularity is a word from astrophysics referring to a point in space-time where the rules of ordinary physics no longer apply. Kurzweil has correctly predicted the growth of information technologies. A quote from the Time magazine article, "In Kurzweil's future, biotechnology and nanotechnology give us the power to manipulate our bodies and the world around us at will, at the molecular level. We ditch Darwin and take charge of our own evolution." The question is, who is we? And what is at will? Whose will?
Well, as I look at the desiccated human blood cells on my car, I become painfully aware that it is not my will, but my enslaver's will.